The overall goal of the following procedure is to obtain super-resolution fluorescence images that elucidate important aspects of vesicle organization at a resolution otherwise unattainable with fluorescence microscopy. This is accomplished by first transfecting hippocampal neurons with DNA encoding photoconvertible chimeras of vesicle cargo. In the second step, sparsely sampled raw images are collected with a super-resolution microscopy system. Next, the raw images are processed to produce a super-resolution image. In the final step, the data is quantified to elucidate the organization of the vesicles. Ultimately, line profiles of puncta intensity can be used to compare vesicle separation to vesicle width, thereby determining whether the vesicles are close enough to generate a cluster.
Using this procedure, single-color photoactivated localization microscopy, or PALM, can be used to study the organization of organelles in most types of cultured cells.
To launch the super-resolution imaging system for image acquisition, begin by turning on the power to the arc lamp. Next, flip the system PC and component switches on the power remote switch to on. After the microscope control tablet comes on, turn on the computer and launch the software. Expand the boot status window to verify that the system is loading normally, and choose the Start System option in the Login dialog. To position the sample for visualization, open the front and top panels on the laser safety cabinet and tilt the transmitted light arm to access the stage and the objectives. Next, put oil on the